Okay, hello again, everybody. We are going to be talking about section 4.4 in the book now, where we kind of left off a little bit toward the end of the previous lecture. So I'm going to, again, share my screen and go right to the PowerPoint that introduces this concept referred to as cellular respiration. This does not really refer to cells breathing. A lot of times students, I think, look at the word respiration and think of, exhalation, inhalation, that kind of thing. But this is really not that process at all. It's a multifaceted, complex mechanism that cells undergo constantly as they try to extract energy from organic molecules. Now, again, think back to chapter 2. You studied organic molecules. Those were carbohydrates, lipids, proteins, nucleic acids. Those are the four major classes of organic molecules. And as organisms like yourself break down food in the form of those four major organic compounds, energy is extracted and made available for cells to use. In other words, this gets back to the idea of cells being able to make ATP, adenosine diphosphate, or triphosphate, excuse me, to permit them to do cellular work, like muscles can contract. Ions can be pushed against a concentration gradient, right? Active transport. Sperm cells can swim with their flagellum. That takes energy. So how do cells harness that energy from food? That's what we're going to talk about in this next section, 4.4. As it mentions here in the box, it is a process that involves literally hundreds and hundreds of different chemical reactions, each catalyzed by a specific enzyme. We're not going to learn all the enzymes, but if you ever were to take an advanced biochemistry course, you would learn all the different enzymes that in different parts of the cellular respiratory process. But we're not going to worry about doing that. But you should have an appreciation that each of these chemical reactions that we will just kind of show you all make use of individual enzymes. So this is, as it indicates, an overview of the cellular respiration process. And what we have here basically is a diagram of a cell. Here's the cell membrane on the very outer edge of this rather columnar shaped cell. This is obviously just a generic artist sketch. But out here is the cytoplasm or cytosol, where we would have various organelles, mitochondria, lysosomes, Golgi bodies, nucleus, and so forth. In fact, here we are depicting a mitochondrion. And you might say, well, why are we talking about it? Well, because remember, what is the mitochondrion generally thought of as? The generic term we think of when we think of the role of the mitochondrion is powerhouse, right? It is here that the cell generates lots and lots of what molecule? ATP, right? Adenosine triphosphate. So that's why it's mentioned here. The other thing that's shown are the three major steps of aerobic cellular respiration. And I'll talk more about what aerobic kind of means and gets at in a few minutes. But I want to just point out the three major stages or parts, and they occur in a particular sequence, which is, in this case, top to bottom: glycolysis, citric acid cycle, or some books will call it the Krebs cycle, named after Hans Krebs, who was, I think, a European scientist who won a Nobel Prize, I believe, in chemistry for discovering this series of chemical reactions. So it's named after him. But more times than not, most textbooks call it the citric acid cycle, because citric acid is the first compound made in this about a 6 or 8 step cyclical set of reactions. And then finally at the bottom here in orange, the electron transport chain. So we're going to talk about each of those, and we're going to kind of discuss how a cell can take a molecule like glucose here at the very top, which you know is a monosaccharide, and from it break it down and extract energy. That's the whole idea. How do cells make ATP? You can come back to this summary slide after you've had the lecture here, and this is going to make a lot more sense. Right now, it looks pretty daunting and confusing, I'm sure, and there's no reason why it shouldn't, but we will go through each of these steps, and as I say, if you want a good overall summary slide, this would be a good one to look at after you've had a chance to kind of digest some of this. So let's start off with the first of the three parts of aerobic cellular respiration, and it's called glycolysis. And if you look at that word, glyco refers to sugar, lysis means to break or tear. So this is the process whereby a cell, your cells, any animal cell, is going to take glucose and break it down. Now, why do we choose glucose? Well, it's not the only fuel that our cells burn, if we can use that analogy, like your car burns gasoline to make it run. Well, your cells burn fuel too to make energy and to do work. We're going to talk about glucose primarily here because it's the primary fuel that our cells burn, if you will, or break down. And you might know from chapter 2 that glucose is often viewed in a ring structure. You might remember from the previous earlier lecture that I gave here in the chapter, you looked at a couple monosaccharides, and they had a six-sided ring. They're called hexose sugars, and glucose is also a hexose six-sided sugar. Go back in chapter 2, there's a diagram of it. We're going to be depicting it, as we see here, as a linear chain of carbons rather than in a hexose six-sided sugar. We're going to lay it out, lay those carbons out. Okay, so these are the 6 carbons. You might remember glucose's molecular formula is C6H12O6. And you might say, well, where are the 12 hydrogens? Where are the 6 oxygens that we know should be here? Well, again, we're not going to worry about those particular elements within the glucose. We're just going to look at the skeleton, if you will. We'll look at the carbon backbone as it's laid out in a linear sort of way here. But we do need to remember that there's more than just 6 carbons in glucose. There's also a lot of hydrogens and some oxygens. Now, this process that we're going to start to talk about occurs in the cytoplasm of all of our cells. So we need to kind of talk about where does this process occur in the cytoplasm. And note that it indicates here in the left-hand column that this can occur in the presence or absence of oxygen. Now, a few minutes ago, you heard me say aerobic cellular respiration. Aerobic means in the presence of oxygen. Most of the time, our cells have adequate oxygen available to them to go the aerobic route, which means they can proceed through all 3 stages: glycolysis, citric acid cycle, and electron transport chain. We go through all 3 steps. If we have no oxygen, we call that anaerobic, and we can only proceed through glycolysis. We can't go beyond that. We can't go into the citric acid cycle or the electron transport chain. So we should just kind of lay that out right now, that particular rule, if you will. Aerobic means go through all 3 steps in the presence of oxygen. If there's no oxygen, anaerobic, we can't go through steps 2 and 3. That has implications as to how much energy a cell can generate. If it has the aerobic conditions available, then it can generate lots of ATP. If it's anaerobic situation that we're dealing with, then we can't get as much ATP being made from the glucose. So let us again take a look at this process. They're showing 3 phases. In reality, there are 10 separate reactions, each catalyzed by a given enzyme. Again, as I said earlier, you're not having to worry about the enzymes. You're not even having to worry about these intermediary compounds listed here, like the fructose 1,6-diphosphate or this dehydroacetone phosphate. Don't worry about the names of those. Don't get hung up on the chemistry. but I just want to kind of walk you through what's taking place basically here in glycolysis. As we take glucose, glyco, and we cut it, the cells begin to break it apart. So in this first phase here that they're describing, look what's happening. The cell is degrading 2 ATPs. Now you might remember from earlier, when ATP is broken down, the end products are ADP, adenosine diphosphate. Go back in the earlier lecture, Zoom show, and you'll see a slide on that. Or you can simply reference page 128, figure 4.8 of your book. That also has it. And we also produce 2 phosphate groups, one from each of the ATPs. We pulled that terminal phosphate. Remember I told you I had a wavy line, a wavy bond there? That means unstable. So we're taking that terminal phosphate group off the ATP, leading to ADP, and we're putting it on the glucose. So one phosphate group gets slapped on this end, the other phosphate group from the other ATP gets slapped on the opposite end, and then of course here's our 2 ADPs. Obviously this has a different chemical configuration, which is why it's called something besides glucose, but it has 2 phosphates. Then basically what occurs in another series of about 4 or 5 reactions is the cell will break this bond right in the middle of the 6-carbon backbone. So right where you have my little arrow there going up and down, we cleave that bond. We do some more modification, if you will, of some of those molecules, and we're basically left with 2 3-carbon compounds, as you can see here. Here's one, here's the other. Then what happens is we introduce phosphate into the reaction, and we're taking now 4 adenosine diphosphate molecules, and we're going to be creating, the cell will create 4 ATP molecules. So it's just the opposite of what happened up here. Here we pulled 2 phosphate groups off, one from each ATP. Now we're adding enough phosphate into the system so that we can take these 4 ADPs, phosphorylate them, that's the fancy name for acquiring a phosphate group. So the ADP is phosphorylated and the cell makes 4 ATPs. So it broke down to get the ball rolling, if you will, but it more than makes up for that toward the end of glycolysis by making now 4. So there's actually a net gain of 2 ATPs, isn't there? 2 from 4 is 2. The other thing that happens here toward the end of glycolysis is the conversion of this molecule that you see here as indicated NAD with a little superscript plus. I'm not going to get into the chemistry of that. I'll tell you what that stands for if you're interested, which I'm sure you're not, but I'm going to tell you anyway. NAD stands for nicotinamide adenine dinucleotide. It's a mouthful. It's a pretty large molecule. And what I'd like you to do is to think of NAD plus as an electron carrier, because that's really what it does. It is a coenzyme that can carry electrons. To try to simplify this and to give you some bit of an analogy or metaphor, because this is really, 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 really important, I would like you to think of NAD plus as the empty school bus or empty bus, devoid of passengers. The passengers will be the electrons. We'll use that analogy. So here's the empty school bus, NAD plus. When we introduce passengers, these folks are waiting to get on the bus. When we add electrons to NAD plus, look what we make. In this case, 2 NADs become 2 NADHs with some additional hydrogen ions, which are really protons. Now, once we add those passengers in the form of electrons onto the empty bus, here is our full bus. Here's NADH. This is the full complement of passengers, obviously, on the bus. Those are the electrons. And you might say, well, where did these electrons come from? The electrons came from these molecules that are part of this glycolysis set of reactions. So they don't come out of nowhere. They are actually extracted from the molecules here within the glycolytic pathway. Ultimately could be traced back really to glucose if you go back far enough. And then here we are at the end of glycolysis. We end up with two pyruvic acid molecules, they're called. And note here, each of these has 3 carbons. So when we take 2 times 3, we get 6, which accounts for what we had here at the beginning, right? This is C6H12O6, glucose. And so we're basically left with 2 3-carbon molecules called pyruvic acid or pyruvate. Some books and videos will use that term, pyruvate. If you ever hear it, it's the same thing as pyruvic acid. And if we had aerobic conditions present, as I mentioned about 10 minutes ago, then we can continue on to the citric acid cycle, which was step 2, and eventually electron transport chain, step 3 of cellular respiration. So that's aerobic cellular respiration. If we have anaerobic conditions present, and we'll talk more about anaerobic conditions when we get to the muscular system, we cannot go into citric acid or electron transport. So you might say, well, what happens to the pyruvic acids here then? Well, it depends on several things, but let's pretend it's your muscle cells, okay, that have run out of oxygen. Let's say you're running a marathon, you're running a race or what have you, and the respiratory system cannot keep up with the cardiovascular system in supplying oxygen to your cells. You probably know that if you deplete oxygen or prevent oxygen from getting to cells, they die. Think of the brain, for example. If you deprive the brain of oxygen, you can die. Any cell can die if deprived of oxygen. So the goal here is to not let the oxygen levels get completely at 0 for too long a time. Now, sometimes cells can withstand some limited anaerobic conditions, and that's what's going on here. So in your muscle, if you can't get enough oxygen into the blood to be taken to the muscles will be forced to go the anaerobic route. And what they basically will then do is they will produce the pyruvic acid, right? Because remember, this can happen both aerobically and anaerobically. So if it goes to pyruvic acid, the only route it can then go from that point forth on forward is, in the absence of oxygen, would be to be converted. These pyruvic acids get converted into what are called lactic acid molecules. And in order to do that, we're going to have to take some of those electrons that we've just stored in the form of NADH, and we're going to have to tell the passengers, hey, electrons, you need to get off. So we can form our empty buses here, right? And then those electrons actually get added to the pyruvic acid and make the lactic acid. Okay, so the lactic acid is different chemically from pyruvic acid. It has some additional electrons in it, basically. This material, lactic acid, can build up in your muscles and give you muscle cramps. And I think we've all experienced that probably at one time or another in our lives. Okay, so to summarize, glycolysis involves the degradation of glucose into 2 3-carbon pyruvic acid molecules. And along the way, the cell will degrade to ATP, make 4, and come out ahead, therefore, 2. The cell will also generate 2 NADH molecules, these electron carriers. We got 2 big bus loads of electrons that we've produced here in glycolysis. We're ready now to continue on the aerobic branch to the next 2 steps. Let's watch a short video here that talks a little bit more about and reviews glycolysis.
Cells derive energy from the oxidation of nutrients such as glucose.
Turn up the volume a little bit.
Cells derive energy from the oxidation of nutrients such as glucose. The oxidation of glucose to pyruvate occurs through a series of steps called glycolysis. The energy released during these oxidation reactions is used to form adenosine triphosphate, ATP, the energy currency of the cell. The initial steps in glycolysis are the additions of 2 phosphates to the glucose molecule at the expense of 2 molecules of ATP. The result is a 6-carbon sugar diphosphate molecule and 2 low-energy adenosine diphosphate molecules, or ADP. This 6-carbon sugar diphosphate molecule is then split into 2 3-carbon molecules. Each of the 3-carbon molecules is converted through a series of steps to pyruvate. During these reactions, electrons are transferred to the coenzyme NAD+ to form NADH, and ATP is formed. Under aerobic conditions, the pyruvate is further oxidized to yield more ATP, and under anaerobic conditions, the pyruvate is converted into lactic acid.
Okay, great. Now, in that video, you may have noted that it used the term pyruvate instead of pyruvic acid. So as I said earlier, don't let that throw you. Pyruvic acid and pyruvate are the same thing. And a cell will generate 2 of those for every original glucose that it started to degrade at the very beginning of glycolysis. Okay, so back to the PowerPoint. So we've got these 2 3-carbon pyruvic acid molecules, right, that the cell has generated. What are we going to do with those? What's the cell going to do with those? Well, it's going to convert those into
Cells.
Oops, sorry about that. It's going to convert those into some additional molecules. So here we are at the top. Remember, this diagram depicts a cell, and inside of it, the mitochondrion, the powerhouse organelle. Glycolysis, we just said earlier, occurs in the cytoplasm. That's why we have it listed out here in the lighter yellow zone. What we're going to do now is we're going to have those 2 3-carbon pyruvic acids move into the powerhouse organelle, the mitochondrion. Okay? And again, we are assuming aerobic conditions are present. Before we go through this series of reactions, I would like to also take a moment and show you some structures of the mitochondrion that I've depicted here in the lower left-hand corner of the screen, because I think it's really critical that you understand some of these different structures. We've already defined the outer cell membrane or plasma membrane, obviously right here, and then as we go into the cell and encounter the very outer edge of the mitochondrion itself, this is called the outer mitochondrial membrane. So that's this in kind of dark orange. As we proceed into the center, or into the interior, I should say, of the organelle, we see something called the inner mitochondrial membrane. So I'm going to kind of just trace that with my cursor. So this is inner mitochondrial membrane. And you'll notice that it has these invaginations here, doesn't it? What do you think those do? From strictly a physics point of view, what are these cristae allowing? Well, they're increasing the surface area, right? That's what they're doing. And you'll see why that's critically important a little bit later. So we have these cristae, these kind of infoldings, if you will, of the inner mitochondrial membrane that function to increase the surface area. And between the inner mitochondrial membrane and the outer mitochondrial membrane, this zone here sort of in pink is called the intermembrane space. Inter means between. So indeed it is the space between the outer and inner mitochondrial membranes. Critically important that you kind of understand what those structures are. They'll have a big role to play when we get into the electron transport chain. Okay, so back to citric acid cycle. The 3 carbons here of pyruvic acid are shown. There's also hydrogens and oxygens there, but we just don't worry about those. We're just looking at the bare bone skeleton, if you will, of this pyruvic acid molecule. It's entering now the center of the mitochondrion called the matrix region. So we have the cristae we talked about earlier, and this zone here in yellow is called the matrix. The 3-carbon compound is broken down into 2-carbon compounds, one here and shown, called acetic acid. And when we pull that 1 carbon off to create the 2-carbon acetic acid, that goes into the production of carbon dioxide. Now, what do you know about this? You're exhaling it right now, aren't you? You know it's a waste product of our cells. I hope you do anyway. Well, this is where some of that is made, right here in this reaction, CO2. In addition to forming a smaller 2-carbon compound here from the original pyruvic acid, look what else is generated. The empty big school bus has acquired some passengers in the form of electrons to form NADH. This is the full school bus. This is the electron carrier that we're going to talk more about later on. We made some already in glycolysis. We're adding to our caravan now. We're adding a 3rd bus to the 2 we made in glycolysis. These, this 2-carbon compound called acetic acid is chemically modified a little bit into what's called acetyl coenzyme A. I'm not going to get into what that is, but basically something called coenzyme A gets added to acetic acid to make this acetyl coenzyme A. And then this 2-carbon compound combines with a 4-carbon compound that was made at the end of the preceding cycle. And when you add 2 carbons to 4 carbons, what do you get? 2 plus 4 is 6. This is a 6th carbon molecule called citric acid. Again, that's where this gets its name. It's the first compound made in the citric acid cycle. This 6-carbon compound undergoes a series of chemical modifications and alterations. Again, not important that you worry about the names of all these intermediary compounds. Don't worry about that. But let's just follow what happens to the carbons here, and if we make any interesting things along the way. So here's our citric acid molecule, gets converted into another 6-carbon molecule, and then we pull a carbon, off this isocitric acid, giving us a 5-carbon now. Remember, 6 minus 1 is 5. And that carbon that we pulled off here, just like up here, goes into the formation of another carbon dioxide, which again is what you're exhaling right now. In addition to reducing the number of carbons here from 6 to 5, look what else is going on here. NAD plus has acquired electrons. We say it's become reduced. That's the chemical term. When a reduction reaction occurs, it has acquired electrons. So NAD plus is reduced into NADH, another big busload of passengers, right? Adding to our caravan, now we've got 4. And then this 5-carbon compound gets converted into a 4-carbon compound. When we pull that carbon off, it goes into another CO2. So we're generating quite a bit of waste product here, aren't we? And we make another NADH from NAD+. So we're really adding the passengers, aren't we, to the big school bus. This 4-carbon compound gets converted into a different 4-carbon compound, and the cell generates an ATP directly. So here's ADP becoming phosphorylated to make ATP, adenosine diphosphate plus a phosphate, adenosine triphosphate. This is that high energy currency molecule that we want to be making. So we've made now 1 here so far. We had made 2, right, in glycolysis. Now is another one. This 4-carbon compound gets converted into another 4-carbon compound. And we see the introduction of a new substance here. What the heck is this? Well, you see where it says FAD gets converted into FADH. Okay, FAD is a close cousin to NAD+. What do I mean by that? What I'm trying to get you to think about here in terms of an analogy is if NAD+ was the empty big school bus and NADH was the full big school bus, then FAD+ is the empty shuttle bus, which when it gains passengers in the form of electrons becomes FADH2. That's our fully loaded but smaller capacity bus. I looked for a shuttle bus. This is the best I could do. Looks like it's somewhere in Thailand. I don't know. But we've got some passengers in this smaller bus. So it can carry electrons just like NADH can, but it can't carry quite as many. That's the way to think of it for now. And then this 4-carbon compound gets modified into another 4-carbon compound, which gets modified into another 4-carbon compound. And again, look what's going on here on the side. Another NADH is formed. So another NAD+ becomes reduced. The electrons come on board to make the full bus of electrons. And now we're back to the finish molecule of that cycle. And if there's more pyruvic acid being altered here into 2-carbon compounds, then these two can combine with the 4 we just made at the end of that cycle to form another 6-carbon citric acid, and we can continue to crank through the cycle, right? As long as we have incoming materials here at the top, we're always going to have available materials toward the end of the preceding cycle to get us into the next cycle. So in essence, the citric acid cycle allows the cell to crank out lots of NADH and also a couple FADH2, in this case only 1, while we're making 3 here NADHs. We also made 1 here at the top. This technically is not part of the citric acid cycle because it kind of leads into the cycle, but for simplicity's sake, I think what we'll do is we'll probably just include this as part of the citric acid cycle just to make it simpler. But technically, this is the cycle down below. This is leading into it. So to summarize, for every pyruvic acid from glycolysis that enters the mitochondrion, enters the matrix of the mitochondrion, what will be made? Well, the cell will generate 1, 2, 3, 4 NADHs, big school buses loaded with electrons, 1 FADH2, smaller shuttle but full, and 1 ATP. It's probably also worth noting that in addition to those major byproducts, the cell is also going to generate some waste, right? We talked about that. In which case we've got a carbon dioxide made up here and actually 2 of them made in the citric acid cycle. So this is, again, a waste product that all cells make that finds its way back into the blood and you exhale it through your lungs. Let's watch a video on this.
During glycolysis, glucose is broken down to pyruvate. A 2-carbon fragment of pyruvate is used to form acetyl-CoA. The acetyl-CoA enters the Krebs cycle, which occurs in the mitochondrion. During the conversion of pyruvate to acetyl-CoA, carbon dioxide, CO2, is produced and a molecule of NADH is formed. The 2-carbon acetyl portion of the acetyl-CoA is transferred to a 4-carbon molecule, producing a 6-carbon compound. The CoA carrier molecule is released. Carbon dioxide is then released from the 6-carbon molecule, forming a 5-carbon compound. In this step, hydrogen is removed and transferred to NAD+ to form NADH. Next, a second oxidation and decarboxylation occurs. Again, NADH and carbon dioxide are produced. In addition, a molecule of ATP is produced. As a result of these reactions, a 4-carbon molecule is formed in the Krebs cycle. Finally, the 4-carbon molecule is further oxidized, and the hydrogens that are removed are used to form NADH and FADH2. These reactions regenerate the 4-carbon molecule that initially reacts with acetyl-CoA. Each glucose molecule is broken down into 2 pyruvate molecules during glycolysis. Then each pyruvate is converted to acetyl-CoA, which enters the Krebs cycle. Thus, for each glucose molecule, the Krebs cycle must complete 2 circuits to completely break down the 2 pyruvate molecules.
Okay. I want to take a moment here and stress what the end of the video just said. It said that because you generate 2 pyruvic acids per original glucose, and each of those pyruvic acids we just saw goes through the citric acid cycle once. If you want to know how many times you've got to go through the citric acid cycle per glucose, the answer is 2, because you're making 2 pyruvic acids, each goes through 1 time. Okay, we'll review that a little bit later, but just understand, we're not going through that cycle once, we're going through it twice, 1 for each pyruvic acid, and you generate 2 of those per glucose in glycolysis. Okay. I'm going to begin now with a video, and then we'll talk in a little more detail about what happens during the electron transport chain. But I will simply say from the get-go that we are now in the mitochondrion. We have been, right? And I want you to remember that the citric acid cycle is taking place here in the matrix region, shown here in orange. What is shown in blue? It's the inner mitochondrial membrane. And again, remember, we talked about the increased surface area. These are the cristae, these little pocketings of the inner mitochondrial membrane, where we increase the surface area significantly. Because now what we're going to talk about, or see in the video, is those electron carriers that we have been gathering along the way, the big and small buses, right? Full of passengers in the form of what? Electrons. So we're going to take the electrons now from NADH and FADH2, and those are going to play a pivotal role in allowing the cell to make a lot of ATP. That's the goal here. How does the cell make energy? We've made a little bit, right? We made 1 ATP per revolution through the citric acid cycle, but we're going to make a heck of a lot more now when we utilize those electrons that we've been gathering in the form of NADH and FADH2. So those school buses are going to pull up now, they're going to open their doors, and those electrons are going to get spilled out, they're going to leave, and we're going to see what those electrons do. So let's watch the video, and I'll warn you right now, you may be watching this 2 or 3 or 4 times, and that's okay, because it is kind of a complex process, but see if you can put the pieces of the puzzle together here. We'll watch it together once here.
When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD+ and FAD are reduced to NADH plus H+ and FADH2. In the mitochondria, the electrons from NADH plus H+ are transferred to the electron carrier proteins, and the protons are transferred across the membrane. As the electrons move from cytochrome to cytochrome down the electron transport chain, more protons are carried across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen, the terminal electron acceptor, and water is formed as the product. The transfer of protons generates a proton motive force across the membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
Okay, that was just chock full of lots of things going on, right? That probably didn't make a lot of sense. So what I'd like to do is we'll go through the video. I'll turn the audio off, the narration off, and I'll talk a little bit about what's going on, just to sort of review, and then you can go back and watch this again, and I would encourage you to watch it. So let's start it off. I'll mute the volume, and we'll talk about what's going on here. So just to lay out sort of the players here, you need to understand the anatomy of the mitochondrion. We went over that in quite some detail. So to orient you, this is the inner mitochondrial membrane. Okay? This is the matrix. This is the very center where citric acid cycle occurred. Here's the intermembrane space, and then way out here would be the outer mitochondrial membrane, and then way out beyond the edges of your monitor would be the cytoplasm. And beyond that, the plasma membrane or cell membrane. So you got to understand where we are. So we're zooming in here at the matrix. In the center, here is the inner mitochondrial membrane, the cristae for increased surface area. We can pack more stuff here, and this is where all this happens. Now you might look at this and say, oh, I recognize this. You better recognize this. Phospholipid bilayer. What are these pink and purple and blue guys? They're proteins, right? They're integral proteins. Where have you heard about this architecture before? Well, again, we talked about it. You should know this. The cell membrane or plasma membrane has the very same architecture as the membrane here of the mitochondrion. So what you find if you study organelles is that many of them are membranous. They have the very same architecture as the cell membrane does, i.e. phospholipid bilayer with embedded proteins. Very, very similar. Okay. So let's take our electron carriers that we've been producing, right, along the way, both NADH here and FADH2. This was the big bus loaded with passengers. This was the bus in Thailand, right? Loaded with passengers, but not as many. What these electron carriers are going to do is they're going to pull up two special proteins here in the inner mitochondrial membrane. The doors of the bus open up, the passengers leave. They, again, are electrons. And the electrons are going to spill out of those electron carriers. So let's watch them leave. And when those passengers leave, when those electrons leave, NADH becomes NAD plus, right? NADH becomes NAD plus, which is an empty school bus, isn't it? And FADH2, when it gets rid of its electrons, will become FAD plus. And these can then go back and pick up more passengers in the form of electrons. They can go back to glycolysis, or they can go back to citric acid cycle and pick up more electrons. So these are nothing more than shuttles. They shuttle the electrons ultimately from different substrate molecules in these different glycolytic and pathways of the citric acid cycle, and they're all taken here to electron transport. And so we've seen those electrons kind of coming off in the form of hydrogen. And what ends up happening here is, and there goes our FADH2 giving off its electrons. What we're seeing happening here are two things. The electrons are shown here in red. They get passed from protein to protein to protein within this inner mitochondrial membrane. As those electrons move from protein to protein to protein, as you're seeing them doing now, that sets up a situation whereby hydrogen ions get pumped from the matrix into the intermembrane space. And you're seeing that occur. These blue spheres with hydrogen ions, they're going to start to accumulate in the space as the electrons get passed from those electron carriers to special proteins. There goes some more hydrogen ions across the inner mitochondrial membrane as a result of those electrons getting shuttled from protein to protein to protein to protein. That's important to understand because ultimately those electrons, I'll back up here on the video a bit, the electrons here that were originally in the buses, right, the school buses, those electrons will eventually combine with some oxygen and some additional hydrogen to make water. So water is actually a byproduct of aerobic respiration. You get water from drinking liquids, right? And you have to do that, otherwise you can die. But what I'm suggesting here is that cells manufacture their own water as well. This is the same stuff that comes out of your tap. Okay, isn't that interesting? That cells generate this too, so your cells can make water. But we don't really focus on that aspect of this whole aerobic respiration process, but it's interesting to note that the final electron acceptor is said to be oxygen, and the end result is the formation here of some water. But what about this energy that I keep talking about? Where are we making energy? Well, remember, we are starting to accumulate lots of hydrogen ions here in the intermembrane space as a result of the shuttling of electrons between various proteins here. So where do those hydrogen ions go? Well, as they accumulate here in the intermembrane space, they would love to diffuse back into the matrix. And they can do that if they pass through a special integral protein shown here in pink that has a special name. It's called ATP synthase. And as those hydrogen ions move back into the matrix, as they're doing here, look what happens to ADP. It becomes phosphorylated. And when you phosphorylate ADP, you make ATP. So there we go. We're making some energy. We're making lots and lots of energy thanks to those hydrogen ions moving back into the matrix from the intermembrane space. How did they get there to begin with? They got there, they got pumped across as a result of the electrons getting passed to special embedded proteins here, which ultimately makes their way to oxygen to make water. But the hydrogen ions, they would love and do diffuse back in to where they came from. And as they do so, the ADP becomes phosphorylated. We make ATP. That's the takeaway message. Okay, so this is the enzyme, which is really a protein here that helps to catalyze that reaction to phosphorylate the ADP as the hydrogen ions move back into the matrix to make high energy ATP. That's what we want to talk about here. We want to not lose sight of the fact that this is an energy producing process. Really critical. Now, for every NADH molecule, for every big bus load of electrons that dumps off its cargo, if you will, in the form of electrons to those embedded proteins, and then the hydrogen ions get pumped across, right, into the intermembrane space, and they come back through diffuse back into the ATP through the ATP synthase into the matrix. For every NADH that does that, the cell generates about 3 ATP molecules. For each FADH2, remember the smaller shuttle bus in Thailand, for each of these that dumps off its electrons into electron transport, the cell generates less than that, about 2 ATPs. So big school bus generates the maximum energy, the smaller shuttle bus creates less energy. Still makes energy, don't get me wrong, it's important, but not as much. So here's a question I'd like you to ponder. How many ATP molecules can a cell generate aerobically for every glucose molecule it started with? In other words, here we have that summary diagram you saw earlier. Here's our glucose, here's glycolysis, here's pyruvic acid, here's citric acid, here's electron transport. How many ATP molecules will a cell generate? It says right here, doesn't it? But can you figure it out mathematically? Well, you should be able to. Let's walk through the steps. Let's summarize what we have in glycolysis in terms of what's being made. I'm not worrying about all the different reactions. I'm not concerned with all the names of the intermediary products or anything like that. I just want to know, what do I start with? What do I end with? What do I make along the way? You can go back and review that, but here's a summary. The cell made 4 ATPs, but degraded 2 early on, resulting in a net gain of 2 ATP molecules. So we were starting off with 2. We're going to be making a heck of a lot more very shortly. Remember, we also generated 2 NADHs in glycolysis. When they go to the electron transport chain, as we said a moment ago, on average, each will generate about 3 ATPs. So 2 times 3 is 6. This little squiggly line means approximately 6. Not exactly 6.0, it's around 6, maybe a little less than that actually. This ETC stands for electron transport chain. So there we go. That's basically what we've got going on in glycolysis. And then those 2 pyruvic acids each enter separately into citric acid cycle. And what do we make in citric acid? Well, for every revolution for each individual pyruvic acid, here's what's made. 1 ATP directly, 4 NADHs. We're including again the 1 that was in the original reaction, plus we're adding it to the 3 in the actual cycle to give us a total of 4. If each of those on average generates 3, then do the math. 4 times 3 is 12. Again, squiggly line means approximately. So those 4 big busloads of electrons will yield 12 ATPs approximately in ETC. The smaller school bus here, the FADH2, remember, can only yield 2. And we made 1 of those per revolution. But don't forget, we've got to go through this 2 times because we make 2 pyruvic acids per glucose. In essence, then, we must change all of these numbers. We have to multiply all this by 2. So now we've got the 4 NADHs, excuse me, that become now 8, yielding 24 ATPs. And then we have the 1 FADH2 multiplied by 2, giving us 2 FADH2s times 2 each gives us 4. Plus we made 2 directly. So when you tally all of this up, the ATPs made directly and the ATPs made indirectly as a result of electron transport, each glucose molecule, just looking at strictly the numbers, we would come up with something close to 38. However, as I've indicated here in the box and also here in the summary diagram, the actual number is not 38, it's less than that because it's not exactly a 3-to-1 or 2-to-1 ratio in terms of ATPs per coenzyme. And there's other things that we're not going to get into that lower the total number of ATPs made per coenzyme. But nonetheless, we're talking a lot of energy, 32 to 34 ATPs per glucose, compared to if this was anaerobic, we would only generate 2 ATPs per glucose because we can't go through citric acid cycle and we can't go through electron transport. So anaerobically, we're like 1/16, 1/17 as efficient in terms of energy production as we are aerobically. So let's go back now to this half of the ATP cycle that I introduced in the previous video earlier in section 4.4, I believe it was. So this is showing the phosphorylation of ADP into ATP. And we just talked about how that occurs as a result of those hydrogen ions diffusing back through the ATP synthase from the intermembrane space into the matrix, right? Well, that's what's going on now. This is talking about the electron transport chain. This is how that ADP becomes phosphorylated to make our high energy ATP. That's going to be used for work, for cellular work, right? Now, we have just talked about how glucose is the primary fuel molecule for our cells. Glucose, remember, is a monosaccharide and is a member of the carbohydrate family of organic molecules. And when we take carbohydrates and we undergo this catabolic reaction called hydrolysis, again, we talked about that in section 4.1 of the chapter, we're going to make monosaccharides like glucose. And we've just finished spending lots of time talking about how monosaccharides like glucose can continue to be degraded to make lots of energy, some CO2, and some water. This, in essence, is aerobic respiration. But you have to understand that while carbohydrates are used in the foods we eat to be degraded, no question about it, we get energy from organic molecules like glucose, but there are times when our cells may want to take those monosaccharides that result from the degradation of, say, starch and other complex carbohydrates, and maybe we don't want to continue to degrade those in catabolic reactions. Maybe we want to kick into the anabolic pathway mode, if you will, and that's what's depicted on the bottom of this diagram. So there are times when your cells will take the glucose and actually combine a bunch of glucoses together to make what's called glycogen. That's how you store typically excess glucose, at least initially, and you store that in your liver and in your skeletal muscle. Now, this is a limited finite storage capacity. You can only store so much glucose as glycogen. If you exceed that, look what you make, fat. Yeah, and that's not necessarily a bad thing. I mean, animals put on fat in the fall. We're coming up now on the time of year when deer and other animals are really chowing down on nuts and corn and whatever they can find, right? They're really increasing their glycogen storage capability, but they're also producing lots of fat. And they're going to rely on that in the winter months when there isn't apples and nuts and corn and much to eat, right? The other interesting thing that we won't get into much here, but I wanted to mention it, is the fact that our body can also sometimes take monosaccharides, and rather than making other types of carbohydrates or lipids, can actually generate amino acids from them. We chemically modify, the cells chemically modify those monosaccharides to make these guys. And what do you know about amino acids? They're the building block molecules of what other organic compound? Proteins, right? So here we are talking about how cells can make amino acids and eventually proteins, or how they can make polysaccharides, or eventually lipid. So quite a diverse functionality of our cells, depending upon the needs at any given time. This is also a very interesting diagram, and it gets into a bit of nutrition, and I'm not going to spend a ton of time on it, but I do want you to note here this breakfast that we're having. we've got some fried eggs, a couple pieces of toast, some butter. We've got some breakfast sausage. Gosh, I'm getting hungry just thinking about it. We have all 3 of the 4 major organic groups of molecules here, don't we? We have proteins in the egg white and in the yolk. We've got carbohydrates, certainly in the toast. If we put some strawberry jelly on there, there's more sugar, more carbohydrate. And we've also got fats in our breakfast sausage, tons of fat, right? And also in the butter. So in a meal like that, or even any meal that you eat, if you just stop and think about it, if it's a big meal, you more than likely will have all major organic molecules there in those foods you're eating. Think of pizza. Well, your digestive system is going to break those down, right? Proteins broken down into their amino acid building block molecules, carbohydrates broken down into simple sugars, fats undergoing hydrolysis to form glycerol and fatty acids, right? These should sound familiar to you. We talked about those earlier in the chapter. These are all catabolic processes. And we just spent time talking about how glucose can enter glycolysis and citric acid cycle and electron transport, and the cell can generate 32 to 34 ATPs, right? In addition to carbon dioxide and water, right? That's true too. There are other things made. But what this is also getting you to think about is look at the amino acids that are going to result from breaking down the protein in our meal. Those amino acids can be used to make other proteins, theoretically, but we're looking at how these can be chemically modified and enter into a variety of intermediary segments of the aerobic cellular respiratory process. In other words, amino acids can go into the formation of glucose or into pyruvic acid, which in turn can feed into citric acid. Or acetyl coenzyme A, you might remember, is one of those intermediary molecules made just before citric acid. And the same can be applied for glycerol. When fats are broken down, glycerol can be converted into pyruvic acid. Fatty acids can be converted into acetyl coenzyme A, both of which again feed into the citric acid cycle and electron transport. So what I want you to just start thinking about is when you eat a meal that has proteins, carbohydrates, fats, you're extracting energy not just from the sugars. You're extracting energy from all organic molecules via aerobic cellular respiration. And it's pretty cool if you just stop and think about it and study it. Some of you may end up taking nutrition, and they get into more detail about this stuff. Again, it's chemistry. A lot of it's chemistry, which I know you guys love. But you do need to understand some basic chemistry. Otherwise, this is just even more confusing. Okay, I am going to stop here. And our last lecture in chapter 4 will be here entitled Nucleic Acids and Protein Synthesis. So I'll look for that coming up next. Okay, get into chapter 4. Start reading it.